Hello students, now I'll be discussing the bits for the chapter cell cycle and cell division. Question number 1. Cell would normally proceed to mitosis without interruption. Option A, once it has entered the S phase. Option B, once it has entered the G2 phase. Option C, at any time during cell division activity. Option D, none of the above. The question is, cell would normally proceed to mitosis without interruption when cell in the interface will go through the three stages G0, G1 and G2 and S phase. G0 is a phase when the cell doesn't want to continue the cell cycle. It is going to enter the G0 phase, but the cell to proceed to mitosis, it should duplicate its DNA, and the duplication of the DNA is going to take place in the S phase. So option A, once it has entered the S phase, the cell will proceed to mitosis without interruption. So option A is the right answer. Question number 2. G0 state of cells in eukaryotic cell cycle denotes. Option A, checkpoint before entering the next stage. Option B, pausing in the middle of the cycle to cope with temporary delay. Option C, death of a cell. Option D, exit of cells from cell cycle. G0 state of cells in eukaryotic cell cycle denotes. Option A, checkpoint before entering the next stage. No. What are the cells that undergo G0 state? The cells which do not want to divide anymore further, which do not want to enter the next phase of the cell cycle, or the cells some have lost due to accident will undergo into the G0 state and resume back once the signals are received. So, right answer for this question is option D, exit of cells from cell cycle, in order to continue it once the cells are receiving the signals. In this phase, the cell cannot continue with the cell cycle because the protein cyclins are absent. Cyclins are going to mediate from transformation of one phase to another phase. So option D, exit of cells from cell cycle, is the right option. Question number 3. In S phase of the cell cycle, option A, chromosome number is increased. Option B, amount of DNA is reduced to half in each cell. Option C, amount of DNA doubles in each cell. Option D, amount of DNA remains same in each cell. In S phase of the cell cycle, chromosome number is increased. No, here the DNA replication takes place, but the chromosome number remains the same. If there are 2n chromosomes, then after the S phase also the number of chromosomes is going to remain as 2n. Amount of DNA is reduced to half in each cell. No, there is no reduction since the amount of DNA is increased due to DNA replication. Option C, amount of DNA doubles in each cell, is the right answer, because if there is 2C content of DNA, then at the end of S phase the 2C is going to be doubled and it becomes 4C. So option C is the right answer. Amount of DNA doubles in each cell. Amount of DNA doubles in each cell. Moving on to next question, question number 4. Which one of the following forms the spindle apparatus during cell division? Option A, chromosome. Option B, centrosome. Option C, ribosome. Option D, chondriosome. Here, which one of the following forms the spindle apparatus? Because for the separation of chromosomes in further cell division process such as in mitosis, the chromosomes must be held by the structures known as spindle fibers for separation of the chromosomes. So, in which of the following forms the spindle apparatus is observed, or it is formed by which structure? So, option B, centrosomes are going to, which contains the uh, centrioles, and from using the microtubules present in the cytoskeleton of the cytoplasm, they are going to initiate the spindle apparatus. So option B, centrosome, is the right answer. Moving on to next question, question number 5. Cell in G0 phase of cell cycle. Option A, exit cell cycle. Option B, enter cell cycle. Option C, suspend cell cycle. Option D, terminate cell cycle. So we have seen the cells in G0 phase. What happens? It is going to exit the cell cycle. After it completes its G1 phase, if it is not wishing to continue the cell cycle because of the non-dividing phase, or because it is occasionally dividing, or some cells which do not divide continuously throughout their life, such as heart cells and nerve cells, they are going to exit the cell cycle. So option A is the right answer. Moving on to question number 6. During which phase of cell cycle amount of DNA in a cell remains at 4C level if initial amount of DNA is 2C level? So in the previous question we have seen the what happens in the S phase of the DNA. Now they have given the reason. We are supposed to find out what is the title of, or the title for this particular reason. So during which phase of cell cycle amount of DNA in a cell remains at 4C level if initial amount of DNA is 2C level? So option A, only G2 phase. Option B, G2 and M phase. Option C, G0 and G2. Option D, G1 and S phase. If you observe the following options, the amount or the doubling of the DNA is going to take place in S phase of the DNA, and it is going to remain same after the G2 phase also. And then after the mitosis takes place, the chromosome number is going to be varied. So till it enters the mitosis, it is going to have the 2C to 4C content of the DNA. That is, initially when it is 2C, in the S phase it is doubled to 4C, and is going to remain 4C after the G2 phase also. So in the options, it will be in the S and G2 phase, but in the options given we don't have an option which is supporting both S and G2. So we will go to option A, only G2 phase contains the 4C amount of DNA. So option A is the right answer. Only G2 is the right answer. Question number 7. It is difficult to observe individual chromosomes during interphase because the DNA has not been replicated yet, or they have uncoiled to form long thin strands. Or option C, homologous chromosomes do not pair up until division starts. Option D, the spindle must move them to metaphase plate before they become visible. So it is difficult to observe individual chromosomes during interphase. Why? Because here the condensation of chromosomes is not taking place. Okay, however, the DNA, when it is combined with the histone proteins, it is present in the form of chromatin threads or chromatin fibers, which are thin strands of threads which are uncoiled in nature. So therefore can be observed only under electron microscope and not in the compound microscope, and these chromosomes can be observed only during the metaphase stage, not during the interphase stage. So option B, they have uncoiled to form long thin strands. Option B is the right answer for question number 7. Question number 8. Liver cells are highly specialized, but when the liver cell is damaged or parts of it are surgically removed, tissue grows. Liver cells fall into which category of cells? Means liver cells, whenever it is damaged or some part is removed due to surgery purpose, again the liver cells are going to grow. It is because option A, cells permanently locked in G naught. Option B, cells subjected to continually remove. Option C, cells that can be induced to enter the G1 phase. And option D, cannot be determined from the information given. Yes, liver cells, they are not going to be in the G naught state for the permanent period, but they can be induced or to resume back to the G1 phase after they receive certain chemical signals. So option C, cells that can be induced to enter the G1 phase is the right option for this question. Option C is the right answer. Question number 9. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides is termed as option A, cell division, option B, cell cycle, option C, cell synthesis, option D, cell lysis. So, the sequence of events by which a cell duplicates its genome, synthesizes other constituents of the cell, and eventually divides. The cell is undergoing interphase and then the cell division phase or mitosis phase. Together, the events taking place in an orderly manner is known as option B. Cell cycle is the right option for this question. Moving on to the next question, question number 10. Period between two cell divisions is known as option A, replication time, option B, generation time, option C, incubation time, option D, interphase. The period between two cell divisions or the gap between two successive M phases, it is known as interphase. Interphase is a preparatory stage for the next cell division. So therefore it is an intermediate stage between two complete cell divisions. So option D, interphase, is the right answer. Generation time is a doubling time of an organism, means an organism, a cell that takes a total period to complete one complete cell cycle is termed as generation time. Incubation time is nothing but it is a time required to produce the cultures. Desired amount of cultures is known as incubation time. So option D, interphase, is the right answer. Question number 11. During G1 phase, cell grows in size, DNA replication occur, neither divides nor synthesize organelles, chromosomes condense. During G1 phase, option A, cell grows in size. Yes, in the G1 phase and in the G2 phase, the cell continues to grow in size and synthesizing the organelles. Option B, DNA replication occur. No, in S phase, the DNA replication occurs but not in the G1 phase. And option C, neither divides nor synthesizes organelles. The cell is neither dividing nor synthesizing the organelle. Yes, though the cell doesn't divide in G1 phase, but it synthesizes the organelles. So option C is wrong option. Option D, chromosomes condense. In the interphase, there is no formation of chromosomes, so there is no condensation of chromosome is going to takes place in G1 phase. So option A, cell grows in size, is the right option for this question. Question number 12. G naught is an extension of G1 phase, is also termed as. G naught is an extension of G1 phase, is also termed as option A, intermitosis, option B, synthetic phase, option C, quiescent phase, option D, premitotic phase. So G naught is an extension of G1 phase, The cells which are not continuing the cell cycle are going to exit the G1 phase and enter into the non-dividing or undividing phase, where the cell is performing all the metabolic activities but it is not undergoing the cell division process. So that phase is also known as quiescent phase. G0 phase is also known as quiescent phase, but in synthetic phase and pre-mitotic phase, the synthesis of DNA is going to take place, and in the pre-mitotic phase, that is in G2 phase, the synthesis of cell organelles is going to take place, okay, that in preparation for the cell division. But G0 phase or the quiescent phase is a inactive state where there is no cell division but the cells are metabolically active. So option C, quiescent phase, is the right answer. Question number 13. Throughout interphase, dash are synthesized at a relatively constant rate, whereas dash are synthesized only during the S phase. So option A, most major proteins, histones. Option B, histones, DNA replication enzymes. Option C, structural proteins, functional proteins. Option D, most major proteins and DNA replication enzymes. Option A, it is most major proteins and histones, or option B, histones, DNA replication enzymes. So throughout interphase, the required biochemicals are synthesized, right? So it is most major proteins is the right option for the first option, but that are synthesized only during the S phase. What are synthesized only during the S phase is histones. Histones are synthesized during the S phase. DNA replication enzymes are synthesized during the G1 phase but not in the S phase. In S phase, DNA replication is going to take place. So these enzymes are already prepared in the G1 phase. So histones are synthesized in the S phase and major proteins are synthesized in the throughout the interphase. So option A is the right answer for this question. Question number 14. During interphase, RNA and proteins are synthesized in. During interphase, RNA and proteins are synthesized in option A, S phase, option B, G1 phase, option C, G2 phase, option D, in both G1 and G2 phase. During interphase, RNA and proteins are synthesized in S phase. No, in S phase only histones and DNA is synthesized. In G1 phase, yes, biochemicals are synthesized which contains RNA as well as proteins. In G2 phase also it is synthesizing 3 types of RNA and proteins. So option D, in both G1 and G2 phase, is the right option where the proteins and RNA is synthesized. So option D is the right answer for this question. Question number 15. In which phase proteins for spindle fiber formation are synthesized? A, G1 phase, B, G2 phase, C, S phase, and D, anaphase. So in which phase proteins for spindle fiber formation are synthesized? That is in G2 phase. The mitotic spindles or the microtubules assemble at the centriole region to form the spindle fibers. So option B, G2 phase, is the right option for this question. Question number 16. Which of the following statement is true? The cell lies dormant during interphase? No. Though it is called as a resting period, the interphase, the cells are metabolically active. Option B. The cell increases in size during G0 phase? No. In the G0 phase, the metabolic activities takes place when the cells are No longer they are undergoing proliferation, so the cell is not going to increase in size. Option C, the cell increases in size during G1 and G2 phase. Yes, in both the cases, the cell growth is going to be continued in the G1 phase and G2 phase. The cell grows in size and synthesizes the other cell organelles and the biochemicals. So option C is the true statement and it is a correct answer. And option D, the key event of S phase is the synthesis of proteins required for mitosis. No, here particularly histone proteins are synthesized and the DNA replication is going to take place. So option C is the right answer for this question.